Thank you.
Thank you very much. Hanu, welcome.
Thank you. Thanks. Yeah, probably saving the questions at the end is better because I tend to go on tangent.
Good.
And start talking about what's the latest album by Taylor Swift. And so, yeah, good afternoon, everybody. Good afternoon, everybody. Yeah, I expect you guys to be. Thank you. I expect you guys to be so loud that the folks in the next room think that we're having a great party, and they will come gate crashing. That's what I'm hoping for. But thanks for coming. And first of all, I think, are there any volunteers here for the conference, national show? Okay. If not, yeah. Oh, all right. Fantastic arrangements, and I'm really having a great time. Thank you for your wonderful hospitality. Let us give them a big round of applause. Wonderful. I think one of the largest shows ever, and it's fantastic. Great arrangements. And yeah, at WSU, I teach a graduate course, and the duration is 1.5 hours each class, theory, and usually end up talking for 2 hours. So now I'm programmed, if I stand in front of a group of people, I talk for 2 hours. So I hope you brought your sleeping bags with you, and be prepared to miss the reception. Thank you. Go Cougs. Yes, go Cougs. But just kidding. Yeah, I'm going to quickly give an update on ongoing research project on viruses of Dahlia at WSU. As Dr. Miner mentioned, we have been working on this for a number of years and made some progress in understanding what viruses are out there in your gardens, and did quite a bit of a nationwide survey to look at different regions of the U.S. and what viruses are prevalent, so that we can target the management options based on that information. So it took a few years to develop that baseline data. So if you look at any particular commercial edible crop like potatoes or wheat or soybean, that's what typically people do to get a baseline data of what is out there in different states, in different producing regions of the country, right? And then based on that, we essentially identified some priorities for the ADS with the help of the virus team led by Dr. Ron Miner, and one of them is to producing clean plants now that we know what viruses are prevalent in dahlias. Because dahlias are known to be infected by at least 20 different viruses. But some of them are experimentally shown to infect dahlias. That means if you have to manually challenge dahlias with a virus X, and it can infect. Doesn't mean that it's a problem. But so out of these 24 viruses, we found a handful of them that are very common. And so the key step to how do you get rid of them, they're typically a virus infection. There is no cure. There's something like that you can spray and kill the virus. Even though they are on the market, like Lysol and others, they claim kills 99% of the viruses. That's kind of far-fetched, right? So most of the virus disease management is preventive. Avoid, whether it's a potato grower or a dahlia enthusiast, is preventive, to start with the clean material. In case of potatoes, there's a large-scale nationwide seed certification program. So there are labs that provide tissue culture produced virus-tested plant material for potatoes that growers plant in the field, right? A similar principle. So we started a tissue culture pipeline to produce virus-tested dahlias. And so we made quite a bit of progress. So today I'm going to quickly give an update on our understanding of viruses in dahlia, the latest, and also an update on the tissue culture pipeline that we established at WSU to produce virus-tested dahlias, right? Again, thank you, Central States Dahlia Society and the ADS for having me here. And it's so funny that the registration was, full very quickly, and I was a bit late to register. And so Dr. Miner got me in, and I was joking with him that what category should I choose to get in? And then I thought maybe I should say I'm a caterer. So food is so important, so I can get in, So again, most of the research at WSU, big thanks to Mr. Jim Chuey, through his charitable foundation, and he made a significant cash gift to WSU. Typically what most universities, whether it's Ohio State, Michigan State, University of Michigan, or Washington State, typically if a particular donor gives a cash gift, what the university does is creates an endowment. That means the university manages that cash gift, and the income that generates from that cash gift is usually used for a particular research activity. So in this case, ADS and Mr. Chuey, they specified when they gave this money to WSU that it should be spent for Dahlia virus research. Right? So that's how these endowments work. Right? So we have similar endowments at WSU for a professor who works on wheat diseases versus apple diseases like that. So my research program is made possible because of the endowment, the gift from a large gift from Mr. Jim Chuey. Right? And also, I like to thank at the very beginning, the ADS has what you call a virus team led by Dr. Ron Miner, and then others here listed. So who have been very supportive in terms of giving us feedback, helping us identify priorities, providing material for research and whatnot. So again, Dr. Ron Miner, thank you. He has been the kind of a driving force behind most of these research activities. Anybody has been to Pullman, Washington? Okay. So you know what heaven looks like. So I'm a bit biased, so you can tell. So we're in eastern part of the Washington state, all the way across from west, like Seattle. So we are only 6 miles state line from Idaho. Right? And it's all wheat country, actually, eastern Washington, a couple of million acres of wheat. Right? So most of the dahlias, are on the west side of it. So I drive quite a bit between Pullman to western part of the state. So I know pretty much every pothole on Interstate 90. So a very, very beautiful area, surrounded by mountains and rolling hills, a beautiful campus. Again, I'm a bit biased. So this is those who haven't seen my slides, please, if you have seen them, you have my permission to take a nap, but please don't snore, that will hurt my feelings. But this is the lab building where my lab is located, very modern, excellent, fantastic infrastructure, prime real estate. Why? Inside is typical lab, benches and a lot of equipment, but when I see through that window on a Saturday afternoon during school year, we look straight into our football stadium. That's why I say it's prime real estate. Yeah.
Get the best seat in the house.
That's right. So circus comes to town with home football games. So today, the coming week is with our football rivals, University of Washington. So quickly online, the outline of presentation today, we have been doing quite a bit of genome sequencing of the viruses that infect Dahlia, because there is some confusion which viruses are different viruses, which ones are same, because that's extremely important to come up with detection tests. So we should know what is that we are detecting and how important or critical it is for Dahlia health. Right? So that kind of a baseline basic information is very critical for management. Right? And based on the genome sequencing data, we developed a molecular test now that we are using for a particular virus. And then we'll end the presentation with an update on the tissue culture project. So basically, a genome sequencing and then detection test and a tissue culture. Mm-hmm. I must acknowledge my lab group. Top left, Dr. Prabu. He's going to kind of tag team and talk about tissue culture, because I'm going to talk about the easy stuff. Difficult stuff, I leave it to him. He likes challenges. And Olga Mamantova, she's a lab technician with a master's degree. She's full-time involved in tissue culture project. She's originally from Ukraine, and last year, a year and a half ago, she approached me looking for a job, and she has a good technical skills, and I immediately hired her. Dr. Muniba is a postdoctoral fellow. She contributed to molecular testing of Dahlia samples for different viruses, and she just left. And Dr. Gupta, she's a postdoctoral fellow, just started participating in the Dahlia virus project, and currently that's the team at the top. The bottom ones contributed over the years to Dahlia projects. Dr. Zai, she moved, got a permanent job with USDA, federal agency, moved. Dr. Romana got a tenure track assistant professor and she moved. Kristen, she was a lab tech. She got a job with the state government lab and she moved. And Dr. Moyo, he's a staff scientist in Cape Town, South Africa. So all of these folks, their research is either full-time or part-time, contributed to Dahlia virus projects over the years. And received training. And again, your support, especially Jim Chuey's support and ADS financial support through endowment creation. An important outcome is that now quite a few received training on not only Dahlia viruses, but in virology. And now your support is providing future generation of scientists who moved on to bigger and better things. And now they are training students now. So it now has a multiplier effect now. So I keep joking with them, Dr. Zai's student, I say that that student is kind of my academic grandson, Ying being my academic daughter, right? So it's generations now. So that's another important, significant milestone or outcome, if you will, from your support. And so quickly, I'll talk an update about a lot of sequencing that we have done. Zach is here. Zach? Oh, yes. Zach is going to talk about a lot of sequencing of Dahlia genome, right? And what we do is do a lot of sequencing of the viruses that attack Dahlias. So ultimately, both of our goal is to come up with ways to make Dahlia stronger and viruses weaker, right? That is the desirable outcome, right? So different approaches, but common goal. Right? So basically, most of you probably are aware of how the sequence looks like. It's basically a string of 4 letters, A, C, G, T. If you really want to define life at the micro level, life is defined by these 4 letters, alphabet, A, C, G, T, and the combination of these A, C, G, T in different combinations actually defines life as we know it, right? That's DNA, right? Our genetic material, right? DNA. So DNA essentially is a storage medium. Our chromosomes, our DNA is essentially a flash drive or a computer hard drive. It stores this information. But DNA makes RNA. That RNA is the one that makes sense to make proteins that are building blocks of us, of life. Those proteins have various activities that makes us who we are. Our skin color, hair color, height, everything is decided by the letters that are stored in DNA translated into proteins. Right? So very interesting phenomenon, if you will. So quickly, the viruses that infect Dahlias, that are common ones, they come in different shapes and sizes, tend to be very small, and codes for only few proteins. For example, human genome, we code for about 20,000 proteins that have different functions that decide our eye color, hair color, skin color, what have you, right? Whereas these viruses are so tiny, they code for only about 4 or 10 proteins. And they are enough to kill an apple tree, an orange tree, or a Dahlia plant. So they have that so much of potency packed into those 4 or 5 proteins. And they hijack the plant. Right? Same thing, COVID is very topical. It is still around. In terms of COVID virus, it codes for about 20 proteins. That's enough that created a pandemic. Right? So now we know what these proteins do so that they can hijack the entire plant or a human being or an animal. Right? So those circle viruses are the ones that very common in Dahlias. Right? 2 groups, those who are not familiar, viruses are grouped into 2 broad categories, RNA viruses and DNA viruses. Essentially, what does it mean? A given virus can have an RNA as a genetic material that causes disease, or DNA as a genetic material. Whereas for us, it's the DNA is our genetic material, and that essentially codes for the RNA and then makes proteins, right? So there are some RNA viruses that are common in Dahlia. So now we have a pipeline to test and produce virus-free plants that are free of these RNA viruses, right? So, and then there are a couple of what you call DNA viruses. Dahlia mosaic and Dahlia common mosaic. As you can see, the names of plant viruses are, we use international convention is simple English. The virus from which a particular plant is discovered, what is the common symptom, and then you attach the name virus. That's it. So if you find a new virus. So in this case, essentially that's the convention. So tomato spotted wilt was first discovered in sick tomato plants in 1920s. But later it turned out it can infect as many as 1,000 different plant species. Very wide host range. Right? The other extreme is Dahlia mosaic virus, which is a narrow, very narrow host range. Dahlia and different species of tobacco, and that's about it. So viruses differ in terms of how many different plant species they can infect. Tomato spot wilt is one extreme. 1,500 or so different plant species it can infect. Right? So they come in different shapes and sizes, and it's important to know which viruses are there so that we can use appropriate detection tests. And that's what, over the years, we spent quite a bit of effort to understand and develop this baseline of database of viruses across US. Here's a nice comparison. The top one is the virus that infects dahlias, tomato spotted wilt virus. The bottom one, can anyone guess what it is? Yeah, which virus? Sorry? COVID, yes, exactly. So I saved a couple of extra wraps from the lunch, so you guys get that for the right answer. COVID, yeah. So some of them look very similar, viruses that infect plants and animals, humans and animals, but they develop this evolution preference, either a plant to attack plant or animal or a human being, right? So top plant virus, it has RNA as a genetic material. COVID, it's an RNA virus. All it carries is a small piece of RNA, codes for about 20 proteins, and that's it, right? So these are extremely efficient creatures, carry very few genes, but they have that ability to hijack you or Dahlia plant. Right? Now we know how they do that, actually. Quite a bit of information is known, right? So RNA viruses, enough I talked about. So rest of the talk, I'm going to talk about these DNA viruses, because again, a comparable DNA virus of human beings, smallpox, which is eradicated, but smallpox is a DNA virus. Whereas polio, HIV, COVID, SARS, flu, common cold, all of them they have RNA as a genetic material, right? And the reason that we engage in this sequencing of these DNA viruses was to understand, are they evolving? Because they do. All viruses are very dynamic. They keep evolving because of a selection pressure to survive. Survival of the fittest, right? Unless any living organism, I tell in the class that they have to do two things to survive. One is to have sex, that generates genetic variation. Two, they have to evolve. If they don't do either of them, they're going to become extinct because of the environmental selection pressure, right? In case of viruses, they do not have any sexual life cycle. There is no male virus and a female virus to mate. So essentially, viruses replicate themselves, and that's what you call is neither asexual reproduction also, right? So essentially, a COVID virus or a DMV of Dahlia, once they enter a living cell, host cell, they essentially turn the cell into a photocopier. So imagine you put one original on a photocopier and press 1,000 copies, you get 1,000 identical copies, right? So what virus does is basically takes over the living host cell, either plant cell or human cell, and starts making copies of itself. And some of these viruses, once they enter a plant cell, within 48 hours, one virus particle can make copies of a million in 48 hours. Very efficient. So it basically hijacks the host cell. COVID does the same thing, right? Or flu or cold, right? So in case of DNA viruses, we wanted to know if they are evolving, at how fast, and what is the implication. The most useful outcome from that findings is so that we get to refine the detection tests. Because we can then improve or design new probes so that we don't miss these new variants. Probably we all heard about variants in terms of COVID. Soon after COVID started, within a year or two, there's a news that a new variant of COVID was reported from South Africa or from another country. So why is it important to know? So that the vaccines can be developed so that those variants do not escape. So that our immune system does not escape those variants when we are exposed to that. Right? Same thing in case of plant viruses, they continually evolve. Different viruses evolve at different rates. Flu is notorious. It evolves very quickly, very rapidly. That's why we are encouraged to take a flu shot every year. Because CDC kind of catalogs all the variants of the previous year and makes vaccine to those variants, so that our immune system responds to that vaccine that has those variants. Right? Our flu shot is nothing but a dead virus, inactivated virus that we get, flu virus. Right? So in case of DMV, I should keep track of the time. I am holding this to show it to myself. So it's a DNA virus. So there is some confusion because most of you probably heard about there are two DNA viruses in Dahlias, DMV and then DCMV. And there was some confusion about the status of DMV. So a couple of my colleagues, one from Australia and one from France, they looked at some of the DMV sequences and thought that probably it needs some verification in the sense that it does not quite fit into the group of DNA viruses that this DMV belongs to. So the way that virus taxonomy works, whether it's human viruses or plant viruses, there's an international association for taxonomy and classification, and there are working groups of scientists who work on a particular group of viruses. So there are about 12 of us from different countries as a working group who work on DNA viruses. We got together and then said, okay, we'll try to figure this out. And my lab being only lab probably working on these viruses, so we sequenced these DMV again from samples from across the country over the last 2 years, right? So essentially, quickly I will go over it about these two viruses, and we finally kind of came up with a clarification about what these two viruses are in terms of relationship to each other. Right? So are they two different viruses or strains of the same virus? In the sense, you can say whether they are siblings or identical twins. Right? So somewhat loose analogy there. All right? Of course, my apologies to Charles Dickens about the famous novel that I borrowed the title from. But these viruses are not as different as Paris and London in that novel. So essentially, Dr. Prabu here, he's an expert molecular virologist. Here is the DNA on the left side. It codes for about 5 proteins, as I mentioned in the beginning. So that's enough for the virus to attack and damage dahlias. Right? So thanks to, again, Ron Miner and virus team, we got a lot of samples from different regions of the country. So we kind of picked West, Midwest, and Northeast, those regions, and collected all the virus samples and then sequenced the whole genome of these virus, whatever virus is there in those samples, DNA virus, and used a method called polymerase chain reaction. And essentially, this is what the sequence data we get from the sequencing facility, and this is essentially the sequence of the DMV that we are looking at. This is the heart of what DMV is, just a bunch of 4 letters, A, C, G, T. But the way they are arranged, the virus can code for different proteins when they are needed in the life cycle. So that protein expression is highly regulated and choreographed. So the virus uses a genetic switch to turn on to make a protein and turn it off when it does not need. It's a very fascinating process, whether it's COVID or DMV. Extremely efficient because there's a very limited amount of genetic information here. DMV, in terms of the perspective, the DNA size genome is 8,000 nucleotides, 8,000. Human genome, it's 3 billion nucleotides. So much more DNA we have. compared to DMV. But imagine if only a fraction of the human genome size, but it carries all the genes it needs to attack plants, right? So essentially, we kind of looked at all the sequences that we generated, compared them, and yes, there are differences in the sequences from different regions. So virus is evolving on a limited scale, and while most of the genes are conserved, there are point mutations, single nucleotide differences. And it has been shown in other viruses that it can make a huge difference in the virus biology for ability to replicate, ability to cause disease, ability to be spread by aphids, and so on and so forth. Right? In case of other viruses, it's been shown. We don't know that information yet about DMV. But what we know now is that these DMV and DCMV are related, and they're more like siblings or cousins, but not identical twins. But they should be considered as siblings because of the genetic relatedness. Right? So you can see that top, they are grouped, all the DCMV isolates from different states, California, Oregon, Ohio, and DMV all grouped. This is the right half of the cartoon. And they are very different from other DNA viruses. So this phylogeny is a very powerful approach to figure out if two viruses are related to each other and how much is that relationship. As I said, are they identical twins or siblings or cousins, right? You are looking at the genetic distance increasing from identical twins to cousins, right? Similar thing here, we are doing it. So this gave us a very clear idea, and now we are proposing that there are internationally accepted rules, and based on that, we are saying that DMV and DCMV are strains of the same virus. They are not different, but they are not identical either, so they are strains of the same virus. That's the important kind of a finding based on the genome sequencing data. Why should we care about it when it comes to testing and when it comes to reporting, and especially when it comes to trade, whether interstate or international trade? So it's very important to have that clarity so that all countries can follow the same classification and taxonomy when reporting, right? So it's extremely important why we should have this kind of knowledge to make those conclusions when it comes to testing and reporting, right? So we just kind of summarize these findings and submit it to a journal, and it's accepted almost. It's coming out in print maybe, hopefully in a couple of months. That we kind of sequenced everything, and we're finding these important differences and what is the status of those two viruses in relationship to each other, right? And based on the sequence data, again, as I said, important practical application is that we use that to develop a new molecular test. And based on using a PCR, polymerase chain reaction essentially is a technique that amplifies the target molecule in plant sample. Most of us are familiar with, for COVID testing, there are two tests, antigen test and PCR test, right? Antigen test, you can do it at home. PCR test, you have to go to a clinic, and they take a swab, and in a day or two, you get the result, right? So PCR test is more sensitive. It takes a bit more time, more expensive, but it's more sensitive, right? So essentially, we use the same PCR test like for COVID test and extract the DNA. That's what the swab there they take from our nose. They extract the DNA from it and do a PCR using probe specific for COVID virus, SARS virus. So we extract DNA, came with a very quick, it takes a couple of minutes to extract DNA using this method, and then use PCR to, this is an instrument, it's only $30,000, so it's quite expensive instrument. And maintenance contract, it's annually, we have to maintain a contract for servicing. Again, it's provided by Jim Chuey. Thank you, Jim, for providing this machine. And essentially, in a couple of hours, you get the result. Those lines that are going up tells me that that particular sample has the virus. If the line stays flat, that means the sample did not have virus. So it happens in real time. That's why we call it real-time PCR. As the process moves forward, you see these lines increasing or staying flat. So at a given time, you can run about 90 samples at a time. So essentially, what's happening is the target DNA is being amplified, so that is read by a fluorometer. And so as the fluorescence increases, if the virus is there, you see that curve going up. If the virus is not there, it stays flat. So it's pretty straightforward to find out if the sample had a virus, right? Very routine, and I think I'll stop here. Yeah, I think I'll give Prabu 5 minutes to, no. When it comes to time, I'm very selfish. Of course, one trick that I found out to keep the Hanu happy is to feed them ice cream on a regular basis. and pay a little bit of salary, yeah. But they care more about ice cream, I think. Again, all these folks at the beginning I showed, they are the ones who did all the work, and I really like them because they do the work and they let me take the credit. So as far as I'm concerned, it's a win-win situation. They may think differently. But, all right, I'll shut up, and maybe at the end after Prabu's talk, I'll be back to answer any questions. Thank you. So I'm going to use this. Okay. I feel quite powerful.
Good afternoon, everyone.
Good afternoon.
Great. Yeah, thank you for joining us today, and thank you for giving this opportunity for me to walk you through the facilities that we have in the Dahlia Resource Center at WSU. So I'm going to explain what we are doing in the tissue culture facility. So it has two parts. So we do tissue culture. I'm going to call it as tissue culture pipeline. So where I'm going to take, where we take a small portion of the plant, we call it explant, and then propagate it. So this I'm going to call it as tissue culture pipeline. So in this pipeline, we may or we may not get a virus-free plant if the mother plant is infected. Still, there is a chance there we might get some virus-free plant, because viruses are distributed, different viruses are distributed in plant in different ways. We cannot tell. Like I remember many of you, like some of you in the last meetings asking question, if a plant is infected, whether the tuber is also going to be infected. Right. And my answer, as well as those who are having a lot of experience like Ron, their answer is like, yeah, it may not. Most of them have, but there will be some that doesn't have. It's all because of they are distributed differently. So the tissue culture pipeline also might end up in virus-free plant, but no guarantee. Then I have another pipeline called meristem tip culture pipeline. So in this case, we're not using any of the plant material. We are particularly using the plant material that is in the shoot tip. That's called meristem. So which is protected from these viruses. Even if the plant is infected with the virus, this meristem shoot tip is protected from this virus. So we carefully excise these meristem, use these cells, initiate on a plant growth media, and then grow the full plant. And there is very good chance that the plant is now going to be virus-free. So I'm going to walk you through these two steps. So why do I need to do the tissue culture pipeline if I know the meristem is going to work? Why do I need to do the tissue culture pipeline? Because the purpose is micropropagation. If I already have a virus-free plant, I don't need to do this tough thing. Yeah, I don't need to take meristem and then grow and then propagate it. So I have a tissue culture pipeline. If I already have a virus-free plant, I take it through the tissue culture pipeline and propagate it. If I have cultivars where I don't have a virus-free plant, I'll be taking them through the tissue culture pipeline again. Why? Because our meristem culture pipeline needs a lot of shoot tips. For each event, we need one shoot tip. So those which are virus infected, I'll take them through tissue culture pipeline. And then I have a lot of shoot buds. I use them for the meristem culture. So here is our tissue culture facility. Laminar hood, which is important to give a sterile environment. And most importantly, we have all these growth incubators that can provide an optimum condition for the plant growth, especially optimum light intensity, optimal temperature and humidity. And thanks to Jim Chuey for providing us the fund to get these grow chambers. We have 2 grow chambers that was funded by Jim Chuey, and we also got.
Each one is only $30,000.
Yeah, and you need to order at least 6 months before that. They don't keep it ready. They start to assemble it or manufacture it after we order it. So we also got one ultra low freezer that was provided by Jim Chuey to store the materials for a long time. Thanks to Jim Chuey. So here I'm going to show the video of our tissue culture facility. So here we have few laminar hoods where we actually do all these tissue culture work, starting from cutting a stem or dissecting the meristem, everything. And next to that, we have some growth chambers, those that was provided by WSU facilities, as well as the one that we got from got funding from Jim Chuey. And this room we have all the -80 freezers. These are the shakers for doing any microbial growth. Yeah, that's how it looks like. That's just the tissue culture facility. That's how it looks like. So we have a very sterile environment with the UV lights to sterilize the room. So now I'll walk you through the tissue culture process. So the first step is to take an explant, a material from the plant, and it can be leaf, stem, or anything from the plant that we are going to start with. So in this case, we are using the stems that is having axillary bud. So we excise the explant and then sterilize them with rounds of sterilization process that includes bleach and ethanol. And then after sterilization, we plate them on the plant growth media. These plated explants were then incubated on these growth incubators that's providing the optimum growth condition, like optimum temperature and optimum light intensity. And then over time, we start to see the shoot buds are growing. So these shoot buds are moved to the magenta boxes that is having shoot induction media, and we get the shoot growth. And I've also optimized the root induction media with different hormone combinations, and it turned out NAA, which is naphthalene acetic acid, is working well for these dahlia. So we moved those that are in the shoot induction medium to the root induction medium and started getting root induction. And we found that different cultivars take different time for the root induction, and their morphology also looks a little bit different. So this is an example here which is of Barbara's favorite and Marcella Lucy. We moved this to the potting mix and maintained them indoor in the lab conditions for hardening. And then we moved those plants to the greenhouse. An example here is Barbara's favorite in the greenhouse. We did the same for all the cultivars. Yeah, the most important thing, it also produced viable tubers. Yeah, we did the same thing for all the cultivars that we have in our pipeline. The next very important part is the foundation block. Again, this has come from during our discussion with the ADS virus team. So we started this foundation block with popular cultivars of Dahlia. What is the purpose of this? Because here we are going to maintain these popular cultivars in tissue culture, where it will remain virus-free and will be served as a source for producing the virus-free plants. So the list which you see here is all suggested by the ADS virus team, and they provided the material. And for the foundation block, so far we have started all our cultivar that is virus infected. And then the list you see here is just the representative. We started cleaning all these using our meristem culture pipeline. So the virus elimination pipeline is established here. So we do one round of tissue culture, which I explained so far, to first establish in the tissue culture. And using those shoot buds, we use them into this meristem culture pipeline. So here we take an explant and we carefully excise the meristem and place them on a growth media. Then then we get shoots, and then we move them to the root induction media, and then we grow them to a full plant that is now ready to transfer to the potting mix. So these plants, plant that is coming out of the meristem culture, if it is tested virus-free, this will be maintained in the tissue culture itself and will now be used as a source for multiplying this. This will be there as a source for as a virus-free source. And this video here, it shows this is how it looked like when we dissect the media, when we dissect the meristem. This is how it looked like in the naked eye. So we cut the shoot, and the meristem is on the shoot apical meristem is on the top. So yeah, so this is placed below the microscope, but what you are seeing is what is captured through the what you can see in the naked eye, not through the objectives, not through objectives of the microscope.
Here I thought it was just a little clip on the top, you get that fresh stem and a disease plant and root that and you'd be fine, but no, you have to get the micro.
Exactly.
Okay. That's why they just say throw it out. Too much work to go that deep.
Okay, so in the last point, you see something really tiny. That's further examined under the stereo microscope to remove any extra tissue attaching to that, so that those extra tissues might carry some virus. So we carefully remove all the extra tissue and just take the meristem and place them on the plant growth media, and then take it through the shoot and root induction and fully develop a plant and move them to the pots. Or also we maintain them in tissue culture. We maintain both in tissue culture as well as in the growth room. Okay, so so we do a number of attempts, as I mentioned before, to get a virus-free plant. For each cultivar, we do number of attempts. And then for all the plants that is coming out of this meristem culture pipeline, we test them all for the DMV and DCMV, which is the most predominant one right now. And all of these source plant had that virus. So we first test them for DMV, DCMV, and all that is tested DMV and DCMV negative, we screen them for these RNA viruses, and whatever you see in green is all negative, and what you see in red is all positive. So we test them, a lot of plants, and then we get some that are virus free. So I have few examples here I'm happy to share this virus free meristem tip culture pipeline is working. We have few cultivars that we got it cleaned through this pipeline, and I have some examples of those cultivars. Excuse me for being very insane in taking these photos, because I don't know anything about the floral arrangement or how it should be staged. I'm having these photos. Yeah, I'm just having these photos as a representative of the cultivars that I have. Yeah, so we have cleaned Holy Hill.
Are you close to that?
Yes.
Okay.
Okay.
Please.
Yeah, for sure.
We need to allow a couple minutes for.
Sure. So. Yeah. So. I'll just be very quick. So it's then we have ACMP, A Fox, Clearview, Daniel, Just Peachy, Edna C, and these two, Clearview AD and Shival Megan. And this is another video which I took, which I think will be helpful for transplanting. As lack of time, maybe I can skip it. Basically, I'm explaining how to transfer this, how to take out from the tube and plant that so that it can give success. We shared, I guess, around 10 of these test tube plants, which are through this meristem tip culture with Dr. Ron Miner.
I grew 10,
And yeah,
14,
so.
got 14 and killed a few.
Yeah, so he was successfully able to grow them indoor, and after a couple of weeks of hardening them and growing them in indoor, he has planted in his garden. Most important slide is acknowledgement. I acknowledge Jim Chuey Charitable Foundation and the ADS virus team, because they have been guiding us always. They have been giving us money. They have been giving us the material that we are using and guidance. So we cannot thank enough. So I also acknowledge the organizers of the 2025 ADS show for giving me this opportunity. Thank you all.